Okay, I think we start. Good afternoon, everyone. I trust that you're happy with the rooms in the diagram. This is not the case, let me know. And how do I see?
It's a surprise transition. Surprise transition.
So in this session, we have 2 talks and 1 additional talk. That's a virtual talk over Zoom. And the first talk will be by Professor Tamar Keasar. Tamar and I studied in the same lab in our master's degree. I studied that. Tamar studied this. Tamar still studies this. And today she's going to talk to us about the plasticity of social behavior.
Good afternoon. So I do like these, but I like wasps as well. So I'm happy with Elizabeth's talk. And I'll talk a bit about plasticity in social behavior. So we know that the transition from solitary living to group living is a major transition in evolution, one of those listed in the book of Szathmary and Smith. And we also know, and it is well accepted, that insects are a very good system to study those transitions. And this is because they exhibit a wide range of social organization patterns, sociality patterns. And here you can see an evolutionary tree of some of the insect images that show just how variable they can be in their social organization patterns. So here in black, we can see the ants and the termites that actually are not variable. All of them are fully eusocial, but all of the other lines indicate images that vary in their level of sociality. And you might ask why there are so many different colors of those lines. And this is because they exhibit different levels of sociality, and there's a big and very confusing terminology associated with levels of sociality in insects. But what it boils down to is 4 main criteria that are used to classify insects by their level of sociality. One is whether there is cooperative brood care. So that means whether more than one female cares for the young, meaning having, for example, workers that perform brood care in the colony. A second criterion is the existence of overlapping generation, and that basically means that in more social species, the colonies are longer lived, so that several generations coexist at the same time. The third criterion is the division of labor, and the fourth one, that I'm focusing today, is whether sociality is facultative or obligatory. So in some of the species, there may be both individuals living as solitary individuals and other individuals living in groups and colonies, but this is not always the case. And you can see that there is a continuum of sociality, starting with completely solitary individuals, where the answer to all questions is no. And at the other end of the continuum are the eusocial species, such as the ants and the termites, that answer yes to the first 3 criteria, but no to the fourth one. So these are also obligatory. Whereas all those intermediate cases of sociality have some polymorphism, and this is what I'm going to focus on today. But just to give a feeling of what those levels of sociality look like, let's look at both ends of the spectrum. We'll look at the example of solitary living and of eusocial living in bees. And this is an example of a bee that lives in a solitary manner. And actually, the vast majority of the species that we have here in Israel and worldwide leads this kind of life. So an adult emerges in spring, it makes a nest, it provisions the nest with food, it lays an egg, it seals the nest and goes away. From that egg, a larva emerges, it develops the pupa, an adult emerges, and the cycle starts from the beginning. So those insects have no communal brood share. Each female lays her own eggs. They have very little. They have very little brood care after egg laying. All the nests last just for 1 generation, about 6 weeks, and this lifestyle is obligatory. Look at the other end of the spectrum, eusocial living. The best known species among the bees are the honeybees. And if we look at their lifestyle, we do have reproductive division of labor. We have reproductive individuals. These are the queens and the drones. Most of the individuals in the colony are the workers that do not reproduce, but they do care for their younger sisters. So there is cooperative brood care. The colony is very large. It can live for several years, so there's a lot of overlap of generation. There is a division of labor, both in reproduction, but also in other tasks in the colony. And in honeybees, this division of labor is age-based. So as a worker older, it shifts. Okay, in the colony. And finally, this lifestyle is again obligatory. a honeybee cannot exist as a solitary individual. But interestingly, this is not always the case, and many lineages of insects show a diversity of social organizations. And here you can see a few of them that we'll focus on later among the bees. These three lineages are particularly interesting because they have a diversity of sociality forms. And we can zoom in on those three lineages. And if we do so, here you can see those three lineages. Thank you. We see something even more interesting, that within those lineages, we have some sub-branches that have facultative sociality. So they have species that can choose, individuals can choose, so to speak, whether to live socially or solitarily. And we can zoom in on those branches even more. So now we're going to zoom in on those top branches of two genera of bees, Lasioglossum and Olictus. And here are their phylogenetic trees. And you can see that each of these genera is composed of many different species. Some of these species are social, others are solitary, and the ones highlighted in pink are those that have that can live both ways. And this is really nice because having these plastic species can allow us to ask what makes an individual act one way or another. How does an individual decide whether to live in a group or alone? And we can answer this question either through experiments or through observations, and gain a lot of insight by asking those questions about the drivers of social living and cooperation. And this has been done quite a lot in many different insects, and it led to two well-established conclusions about things that may cause individuals to live in groups. Right? So one thing that promotes social living is high genetic relatedness, and that can be achieved in a group either through their haploid genetic makeup or through monogamous mating or by inbreeding. So I'm going to talk about that a little bit. A second factor that can promote group living is when dispersal is difficult. So if there are many limitations or restrictions on new nesting sites, for example, that could push individuals to live in groups rather than on their own. And I have some thoughts that I want to share with you later on about a third mechanism that might also promote social living in those insects, which is food sources that are difficult to handle. But focusing first on the two first mechanisms, I want to show some examples of those two mechanisms at work. And again, there are many examples of experimental and observational studies that looked at those mechanisms. Out of those, I chose to share with you just studies. And the reason I've chosen those is because they were done here in Israel and they looked at local species. So one of the studies looked at social organization in the date stone beetle, Cocotryph. One looked at sociality in carpenter bees, two species of Xylocopa that we have here locally. So let's look at the first story, those date stone beetles. So this is what they look like. They're quite tiny. They're about 1.5 millimeters long, and they live within the stones of dates. They're also a minor damage to dates. And what they do is a founder female digs a gallery in the stone and lays her eggs inside. And the offspring usually remain in the same stone and spend their whole life in it. They feed on the stone material, they extend the gallery, they mate, they lay more egg, and they do so again and again until the whole seed is consumed, and that takes about 6 generations. When there's no more space and food within the seed, there is dispersal. And a generation is about 1 month, so it's about 6 months. So there are some dispersal events. And actually, we can think about those beetles as either living alone, those who would be the dispersers, who are also the founders of a new colony and a new seed, or as individuals that live socially in the seeds, and it's actually an individual decision whether to disperse or not. So they're free to choose. We also know that they have haplodiploid, which is quite unusual for beetles, but we do have it. They're highly inbred. They produce broods with a highly female by sex ratio, and they have communal breeding and brood guarding. So there are some guards sitting here at the entrance to the gallery and keeping away predators. So they have several aspects of social living in a beetle. And the question is, what drives the decision of an individual whether to remain in its natal seed or to disperse? Yes. It's going to lay eggs, and those eggs will hatch, and then it would die. So it won't experience actually much group living. It would act as a founder. Yes, and each of the offspring individuals can again make the decision whether to disperse or to stay. So in a study by Sitkov, Sharon et al., they looked at the 2 hypotheses for what would make an individual stay within a seed, which are kinship, high kinship, or scarcity of resources, and manipulated both. So they created populations of beetles that were either highly inbred or had low inbreeding, so they manipulated the genetic relatedness, and they kept them in conditions with either a lot of seeds available or with a shortage of seeds. Okay, so this is the combination of abundant seeds or limited seeds, and they followed colony development in those 4 treatments. And the bottom line, which you can see here, is the proportion of dispersing individuals in the 4 treatments. So these are the outbred beetles. These are the inbred beetles. This is with high seed abundance, and this is with low seed abundance. And we can clearly see that seed abundance didn't matter to them at all, but relatedness clearly did. So they were more likely to disperse when the relatedness among them was low. They also looked at the implications of dispersing versus non-dispersing and asked, does social living have any benefit for those beetles? And when comparing the number of eggs laid, there was absolutely no difference between the 4 treatment groups, but they saw that in the inbred beetles, there was more social living, more of the eggs survived, and the number of immatures per colony was higher than in the outbred ones. Okay, so this is one story, and that story points to the importance of genetic relatedness in driving the decision whether to live socially or not. No, usually they're highly inbred, yes, but they artificially created non-kin colonies. Okay. So what they could see.
Or I could say, oh, that even individuals aren't good competitors, so they should stay around because they won't make no sex. So there are many potential considerations I'm not very well taken into.
So their observation was that the brood guarding was better in the inbred populations. And they think that this leads to an extra benefit of staying together. Happy with more questions. Okay, let's go on to the next story.
What about resource availability?
So this they did by manipulating the resources, the amount of seeds available. That didn't have any effect. Mm-hmm.
So eventually you're going to deplete the resources within a day.
Within a day?
A day.
Yes, yes. Yes. Mm-hmm.
Don't they have no choice?
Then they have no choice, right?
How do they have choice? That's really what you ask.
Okay. Let's look at the second story. So the second story deals with carpenter bees that dig nests in wood, and actually in some places they are considered also pests because of that. They lay several eggs within the nest that they made. Here you can see how the nests look like, and here you can see the development of the offspring in the nest, and you can see that there are several offspring in each nest, and they're not synchronized. So some of them are more highly developed. These are the older offspring. Some of them, the younger offspring, are not yet equally developed. And, this is the mother that guards the nest. And when the first daughter emerges, reaches adulthood, very interesting dynamics takes place between the mother and the new daughter. And there's several possible outcomes of those dynamics. One is that the daughters leave their natal nest and found a new nest. They can dig a completely new nest. They can chase away a resident from her own nest and recolonize an existing nest, but they leave their natal nest. Or they can fight with their mother, kill the mother, or evict it and take over the mother's nest. Or, in some cases, daughter remains in the mother's nest as a guard. And the nest effectively becomes social with two females guarding, feeding, and taking care of it. And again, this is to me a matter of choice. So a daughter can decide whether to join her mother's nest and live socially, or leave and live solitarily. And we know that the very same individuals can change the decision throughout the season. So there's a lot of plasticity and flexibility going on here. And again, the question is what would drive a daughter's decision to nest socially or solitarily? And this question was very nicely addressed in an old study by Katja Hogendoorn, who observed nests of carpenter bee nature. She had the bees marked, so she could follow the same population over a whole summer. She could make sure that the composition of the population didn't change throughout the summer. So actually genetic relatedness among the bees remained the same throughout the observation period, but resources did not remain constant. In the spring, there's a lot of nesting sites, there's lots of flowering plants, so resources are abundant. This is the first half of the season, which is shown here. And then, as one reaches summer, the resources become much more scarce. There is competition for nesting sites, there is competition for food, especially pollen that the bees feed on. And these are the observations that they made in the second part of the nesting season. And what they saw is that during the spring, most of the bees form solitary nests, whereas towards the end of the season, in late summer, the abundance of social nests is very much. When they looked at the consequences of social nesting, you can see that in the beginning of the season, there is quite little robbery of pollen by other bees who come and steal the pollen, but this increases very much towards the end of the season when resources are scarce, and there's much more pollen robbing in the solitary nests than in social ones, because there's only one individual to guard the stores. Also, takeover by intruders were higher in solitary nesting in the beginning of the season, but the trend was reversed towards the end of the season. So here, actually, it seems that living in groups, a small group of 2 individuals, is actually beneficial when outside conditions, when ecological conditions become more difficult. And genetic relatedness doesn't seem to matter so much. So, as I told you, these are just 2 examples, but there are several more studies along these lines. And basically what we learned from these studies, summing up so far, is that social organization in insects shows a lot of variation. Some of this variation is intraspecific, and experiments and observations on such flexible species can provide us insights about the drivers of sociality, and there is quite a lot of evidence for kinship and resource shortage as drivers of social living. Another take-home message, I think, is that bees are a really nice system to make these experiments and comparisons, both because of the variability between species and within species. Well, so far I haven't talked about our own work, so I would like to do it now. Okay. So when thinking about those drivers of sociality, I also got to think about recent work that we've been doing at my lab on bee behavior. And I think that there are some links between the work that we are doing, which deals with morphologically complex flowers, and social living in bees. And I would like to share those thoughts with you, and I would be really happy for feedback and discussion. So the projects that deals with the question, how do morphologically difficult flowers survive in nature? We know that there is a lot of morphological variation in flower shapes, and flower shapes range from very simple flowers. So these are large flowers, they're bowl-shaped, they're flat, they offer their rewards in very conspicuous locations, and they are very easy for bees to manage, through all kinds of intermediate stages until we reach flowers that are highly complex, that need a lot of manipulation to get at their reward, such as this Darwin's orchid, which which can only be managed by one single pollinating moth. And we also know that there is a kind of game between the preferences of insect pollinators and flower shapes. So if we are looking at a continuum between very simple and very complex flowers, then most flower visitors would easily manage the simple flowers, but much fewer of them would be able to handle and feed from the complex flowers. And as a result, there is much less exploitation of complex flowers, which makes them highly rewarding. So on the one hand, it makes a lot of sense to feed from complex flowers, but on the other hand, it requires higher handling time and higher energy to handle, and also it requires a lot of learning. So we know that when pollinators first approach complex flowers, they don't do very well. The reward impact rate is rather low, and as they gain practice, they gradually improve and learn to exploit those highly complex flowers.
Or is it also about the actual?
It's both less consumption and more production. So to sum this up, complex flowers are highly rewarding to pollinators and bees, but only after practice. Now, if we consider a bee, we can choose among many different types of flowers, some of them simple, some of them complex. Why on earth would they ever visit the complex flowers? Why would they invest all this effort in practicing and learning how to handle them, especially given that the initial rewards are quite low? So we set out to study this question in our lab, and we worked with bumblebees, which are social insects that live in colonies of a few hundred individuals. They don't have flexibility in their social organization. All of them actually must live in the colony. And in one of our experiments, we allowed the bees to choose between flowers that are naturally complex and flowers that we simplified. So each bee was exposed to flowers of one species, either Tecoma, or Antherynum, or Lupinus. Half of the flowers were as nature designed them, and half of the flowers we simplified by moving one petal, so they were much easier to handle. We made sure that the rewards in all of the flowers were just the same. We allowed a single inexperienced bee to approach those flowers and visit them for several dozens of visits, and we recorded their choices, time budgets, and feeding success. We should also note that these flowers are arranged along a continuum of complexity, so these are fairly simple. These are of an intermediate complexity, and these flowers have highly complex shapes. So we saw a couple of interesting things. The first was that when we looked at the very first choices of the bees, all of them preferred the complex flowers somewhat. So we made 60% of the bees for all flower species chose the complex shape. So this is not a very strong preference, but it was highly significant, and there might be some innate preference from complexity going on here. More interesting for this talk were their eventual choices after they gained practice with the flowers. So we found that in all flowers we had actually 3 types of bees. Some of the bees, after practice, specialized on the simple flowers. So they directed more than 60% of the visits to simple flowers. Some of them specialized on the complex flowers. And The remaining ones didn't have a clear preference. These are shown here in gray. You can see that as the complexity of the flower shape increased, more and more bees specialized on the simple flowers and visited them primarily. But still, some of the bees, even if in the most complex shape, the lupins, specialized on the complex flowers. But we videotaped those bees, and we noticed that many of those visits were unsuccessful. So the bees landed on the flowers, they tried probing them, they tried handling them. They didn't manage to get to the sugar solution that we gave them inside the flower, and eventually they left. So when we looked at just successful visits that resulted in feeding, we saw quite a different picture, where actually none of the bees that specialized on the complex flowers here and here, in those two more complex species, ever managed to feed from them. They just tried and tried again and again and again, with no success. But thinking, how is it? How can they manage to just try and fail, never give up? And our thinking is that those bees have a backup system. And the backup system are the other colony members that forage on the simple flowers and do quite well. So they kind of buffer those exploratory bees from their poor foraging success. And our idea is that sociality may buffer against the initial failure of foragers that specialize on complex flowers. And if this is the case, then we have one more driver of social living. If you're a bee, you're feeding on complex flowers, you actually cannot afford to do so if you live alone. Yes. Socially, you can afford to do it and eventually get at those high rewards and increase your foraging success. And this is the idea that we want to explore in our next experiments. Meanwhile, we did some preliminary checks to see whether this idea makes sense at all. So one thing we did was to look at databases of plant pollinator communities, where we have a whole list of plants, a whole list of pollinator species, and their relationships. So we know which pollinator visited each plant, and we can score the morphological complexity of each raw species. We can score the sociality level each pollinator species, and we can ask, do indeed in nature social pollinators visit mostly complex flowers? And this is the correlation that you can see here for just one data set from an alpine plant community. And you can see that those plant species that received mostly visits from social foragers are the complex ones. So these are the high complexity scores, and the complex flowers indeed received most of the visits from social communities. So that's one clue. The other clue that we have is that we are playing with a mathematical model that tries to predict the optimal choice of workers in a colony that can choose between simple and complex flowers. And what our model predicts is that such foragers should start with simple flowers, practice gradually on the complex flowers, and once they learn them, to switch to foraging mainly on complex flowers. So here you can see the proportion of workers in the colony predicted to forage on complex flowers as a function of time. As time goes by, more and more foragers specialize on the complex flowers, but the different symbols here relate to different proportions of foragers within the colony. So if everybody in the colony forages, then specialization in complex flowers is complete and happens quickly. If the proportion of non-foraging bees increases, such as here, then more are expected to remain with the simple flowers, and this is the situation that we would predict, for example, in the case of one solitary nester with a lot of brood in the colony that doesn't forage. These are our ideas so far, and we have the equipment and infrastructure to run experiments with groups of foragers, different colony sizes, different types of morphological complexities of the flowers in our lab, and we hope to explore this idea further in the future. Thanks.
Thank you very much.
There is social learning, observational learning in bees. They learn to find resources by observation. In these flowers, when you're doing the experiment in natural conditions, when you have a high density of flowers, there's more than one forager there, and there will be differences in expertise. So did you take into account social learnings, observational learning in these conditions?
Yeah, so I have natural conditions. Of course, in our lab experiments, we release from the
Yes. Yeah.
in a natural setting.
No. But you can do it also in your lab by releasing an expert bee and a non-expert and a naive one and see what happens. And so.
I have two questions. One is about the facultative species that you described. Are there cases among them where the females are multiply mated or not? Because then Petri lines can be different, and they might have a different genetic propensity for the different decisions, and that might actually maintain some kind of a ESS type of mixed ESS, right? I mean, that would be an interesting thing to look at. Where the decision thresholds are, for example, in behavior. That's the first question.
Yeah, I know that the genetic information exists. I don't know if it has been linked to
And the second question is, I start with an analogy. So in the case of the origin of multicellularity, we have now rather good evidence that a lot of behavior, cellular behavior that is important for the primitive multicellular animals, for example, you find in a patchy distribution in the protists, in the unicellular eukaryotes. And when they are put in the multicellular context, some things you get for free. For example, some of the cells will be inside and they will react accordingly, without any direct selection, and that can be used in the multicellular organism. So, by analogy, has anybody looked at the closest solitary relatives of the social lineages to see which individual behavior elements could have been serving as pre-adaptation to social life? Right? So, what would be a response to some inanimate condition would actually be recruited, and the trigger would be now animate, but the reaction is the same. Everybody looked at this comparison.
It's a great idea because the phylogenies are well known at the same.
Thank you.
Just wanted to notice the very striking resemblance of the story that you told about those individuals who can take a lot of time trying to figure out how to approach complex flowers, based on the fact that they can eat stuff that other members gathered. If you look at the arguments concerning the emergence of big game hunting in hunter-gatherer societies, I think that one of the things you see there, and a lot of people are saying, that if you look at chimpanzees, for example, they will have to stop hunting after a half hour, an hour or two, because if they fail to get anything, they have to still spend many hours feeding themselves in another way, whereas the hunter-gatherer relationship is one in which the hunter can take the risk of failure because there's a different type of food, which means that maybe there's a whole thing going on with respect to risk management that I think we should all probably pursue. It seems to be a very productive way.
Brings me to the last slide that I decided to speak for that constraint. So if we want to make the analogies about humans, should we think about learning difficulties of tasks? Should we think about genetic relatedness? And should we think about resource constraints as drivers of the next level of sociality? I think that this is fascinating to discuss.
Thank you very much. So our next speaker, it does not need a lot of introduction, and I will just say that we have here Eors Szathmary from the Hungarian Academy of Sciences.
Thank you very much, and thank you very much for the invitation. It's extremely good to be here again. I already had a mini workshop in Jerusalem, so an unofficial one that I greatly enjoyed. I'm going to paint you a big canvas, so fasten your seatbelts. Sometimes it will be, at the speed of light because I want to cover an amount of stuff. But I try to be understandable nevertheless. First thing I want to emphasize the following, that some people at least don't somehow take in, that as David Queller in his book review noticed, the major transitions in 95 was actually a synergistic combination of two books, as it were. One was this transitions to higher level units. The other aspect was the origin of novel inheritance systems. And I think that one of the most important messages was how these two aspects are linking to each other. Already, Paul mentioned these two kinds of transitions, also, by the way, identified by David Queller. One is this fraternal type of transition where the units that stay together are related with each other, and there is the egalitarian type of transition where the units are originally unrelated. For example, the mitochondria were clustered in any they were not related either to each other or to the genetic material out of which the nucleus evolved. And it's very important to realize that also, although we a higher level unit of evolution, this must be differently regulated from the fraternal case. There is no way that the mitochondrion could do the reproduction for the plastic or vice versa. It just doesn't work that way. And so therefore you cannot have in the eukaryotic cell, for example, a unique organelle germline, because there are qualitatively different organelles. It's a very simple thing, but one has to think about it. So when you are doing an egalitarian transition, and remember this towards the end of my talk, there are things that are not an option, right? So that's important. Now, there have been several important developments. One of this is this book, which is a gold mine for comparative data and reasoning, but especially for the fraternal transitions, where the idea of kin selection and inclusive fitness, these things work extremely well. But it's a gold mine for that particular aspect. In that book, the author also neatly identifies that cast into different phases. There is this initial group, then there is the social group maintenance, where there are already mechanisms for a more ritualized form of staying together, and this is what he calls which is the ultimate entrenchment, of the transition. Now, people are divided actually. So some people would the major transition line already here, right? Others actually insist that we should be more stringent and we should put the major transitions line here. when you are going through these examples. I'm not going to go through all these examples, but you see what I mean. It's also important that these phases, of course, can take millions and millions of years. So it's not it doesn't proceed at the speed of light. But the phases are different, and remember, authors can differ whether they put it here or here. Now, a few years ago, I have updated the list of the major transitions, and obviously I don't expect you to read this table. I just mentioned that there are interesting developments in the field. For example, in the case of the cells, it could be the case that meiosis, reductive cell division, actually is more ancient than mitosis. That sounds striking, but if you think about it, it actually makes a lot of sense because the ploidy levels were not stringently regulated, but then you have to relieve yourself of very high ploidy levels, and therefore some kind of reduction division was necessary. Also, it's also true that if you look at it, there are about 40 different types of mitosis, but meiosis is almost completely universal, extremely highly individualized. So that's an interesting, I just mentioned this. Now, if it's the case, then the origin of the eukaryotic cell and the origin of sex cannot be disentangled from each other, right? So this is why the origin of sex is not a major transition anymore in this list. It's encapsulated within the transition to the eukaryotic cells. Here you see an interesting column, which is the limited transition space. And those are experiments in evolution where I close to the
karaoke machine. Not used to.
Okay. So what I want to say here is that there are interesting experiments where things look like as if you were to start a major transition or something like that, but the phylogenetic impact, as far as we know today, is still very limited. For example, the origin of soma and germ within the ciliates, with a single cell, is an extremely interesting case, but it was limited to that lineage and basta. Similarly, you can have more amino acids than 20 in certain lineages. You have the 21st and the 22nd, but those are again limited to certain lineages. But these are interesting experiments because then looking at them gives you comparative evidence for other types of transitions. Paul was touching on this topic. So there are two problems usually that you have to solve. One is the cheater problem, when you have something which you could call an altruist and somebody else who doesn't care then, for the good of this local group. The other thing is, which is usually not considered in the classical literature of altruism, where it is not one altruism and one cheater, but you need actually more than two different types of altruists in order to work together, right? That's a different problem. And for a long time, it hasn't been realized at all. Now, these mechanisms work in various contexts. So this is a modified type of Wilson model, where, for example, this one is the good guy and this is the cheater. And the question is whether by transient group formation, you can actually fight against the cheaters. Because if you are just in a well-stored pool, this is not an option. The cheater is the question is whether you need reproductive cells or already transient compartmentation might The answer is actually very clear. It's a bit surprising when you first hear the thing that really makes the difference is whether the altruist can scratch in its own back, right? So, for example, if you have a molecule for which it's true that a single molecule, if it contributes, something, for example, to metabolism, whether the single replicator can feel its own presence, right? Because if it's a replicase, it doesn't work, right? So if you have a molecule of a replicase ribozyme, catalytic RNA, of course it cannot replicate itself because it's one copy. You need another one of itself that's a template. That makes the huge difference whether this simple transient compartmentation without kin selection works or not. That makes the difference. As we also experimented, that you can calculate it. So in a way, from the theoretical trivial paper, the beauty of the paper is that it was done very nicely with experimental methods. So for example, here is the right? This is a ribozyme, which has a catalytic domain. It can perform a catalytic action. It can be replicated. That has to be the invader replicase. And then you see this work now. Of course, cheaters arise because the catalytic domain can just fall out. And then you have a very efficient text sequence which just says, replicate me. Screw it, replicate me. And of course, it's smaller, so it's replicated faster. And as expected, this is why I say, in a way, it's trivial, that if you do it in a batch culture, so the cycle is like this, of course, the parasite is winning. If you, however, make a selection step, whereby you can measure the amount of the substrate conversion, and you just throw away those bubbles that contain not enough substrate produced by the catalytic ribozyme, then you can maintain a stable you have got the cheaters as well, but they cannot kill the system. So this is the transient compartmentation. Also, this hasn't been tested yet. This transient compartmentation is known by our theory to also solve the problem of these synergistic partners, where you need two different types of altruism. So, for example, if you take this case, you know that you have got two different guys, and both of them are important to produce basically anything. So the production of X happens with the efficiency of A, production of Y happens with the efficiency of A. But these groups where it's only this guy or only that guy, this just doesn't work. Then again, you can demonstrate that although in a batch culture, again, if A is bigger than B, X would actually competitively displace Y, even though both of them are necessary for reproduction, right? So this is really a demise, because ultimately, if X takes over, X is not going to be able to provide it, right? And that happens in a Western case, not if you have got local groups, but again, the local groups can be completely randomly formed. You don't need Hamilton for this problem. Again, because one replicator, you know, already one replicator can feel its own presence through the contribution. Now, of course, this is a poor man's form of compartmentation. A much more stringent version is it's actually historically earlier. It's from 687. It was applied to the origin of eukaryotic cell, but also to the origin of protocells. It's called the stochastic corrector model. Here you have the same situation. So these are the two necessary guys, but we are in progenetic conflict because, of course, of course, they can have exactly equal replication rates, but that's a probability measure of 1, right? It's not impossible, but you don't count on it. So therefore, you have got the possibility of internal competition. And what is important is that when you are dividing, you are generating stochasticity. That's one thing. The other thing, which is not depicted here, that even growth is stochastic because you have a limited number of molecules in each compartment, which means that you cannot apply deterministic population dynamics. There is demographic stochasticity with each compartment. So, kurz und gut, as the Germans say, ultimately what you can actually mathematically prove that you will have a stable distribution despite the fact that in every cell, one guy is replicating faster than the other on the condition, of course, that both are necessary. So there is an optimum composition whereby the protocell fitness is highest. The experimental test of this is in the making. It requires the microfluidic machinery that and the other colleagues have developed in Paris, but they know about how to do it. Now, the last thing, of course, you can ask the question, what would be the next step? And the next step is the formation of chromosomes. The formation of chromosomes is a good thing because it's, first of all, it's a conflict mediator, right? It's like a policeman, only one child, if I replicate them, you also replicate, that reduces the conflict between the genes. The other thing is that, of course, once you have chromosome formation, then if one gene ends up in this cell, it will be sure to have at least one synergistic partner in the same cell, but of course that requires that the mutation rate should go down by evolution, because otherwise the genome would be too long. Okay, I'm not continuing. It's important that the chromosomes are an important thing in this kind of egalitarian transition. Now, we also introduced the concept of the filial transitions. It's actually brand new, and it starts with the following idea. As some people put it, when you are in the traditional transitions framework, when you are creating a higher level, you are basically de-Darwinizing the lower level so that they don't, screw up the whole system. Now, there are cases in evolution. One is absolutely clear, the other might also be a valid one. When you are actually de novo Darwinizing something within your system. So, in the case of the classical transitions, you are adding up the new level, so to speak, at the top, right? By terminal addition, to use this thing from anatomy, right? The filial transitions happen by intercalation. It appears within the established hierarchy. And of course, the best known example is the adaptive immune system in the vertebrates. Because in the adaptive immune system, what is happening is basically, it's a combination of two things. This is actually, by the way, Niels Jerman, an extremely important and intelligent immunologist. And that was the title of his Nobel speech, The Generative Grammar of the Immune System. If you are interested in language, you should pay attention to this kind of, it's deliberate why he gave his examples, because what you do is an immune system, you have a generative system where you can generate an incredibly vast space of antibodies, right? Okay, so generation, almost unlimited potential with limited means, that is a number of rules, that is what it means. But also then you select on the basis of the diversity that you are generating, and that's the Darwinian element. It's kind of artificial selection that is happening within your body. It's artificial because it's genetic evolution of the organism that gives the constraints of the selection that is going on within the body. Okay. Now, I mentioned this because there might be another one, which is equally important, and that is related, although not limited to language, to my mind. This is a kind of picture that illustrates the complexity of language origins. And there are here, Paul loves time scales. Here is actually 3 time scales interlocked, So of course, this is the beauty and this is the difficulty of this kind of process. And this is why some of our colleagues wrote an introductory chapter to a book saying that the origin of natural selection, the hardest problem in science, question mark. Maybe not the hardest. There are other minor things like consciousness, unified physical theory, but definitely it's one of the hardest problems. I would actually submit them. Now, there are different approaches, or how you want to approach this. I want to just mention that, of course, you need an ecological setting for these things. And unfortunately, this gentleman deceased about 2 years ago, Derek Picker, from this party with whom we have been working on this idea that primitive language evolved during the time of Homo erectus. It was also related to climate and food change at the time. And it was a highly cooperational ecological constraint, whereby they had to acquire more protein, and the only thing at that time in large quantities was big carcasses. Now, big carcass is big, right? So you You have to find it, you have to inform the others, you have to butcher it, you have to carry it. But that's not enough. Why it is confrontational? Yeah, because the other animals, like lions, and they are standing there and they are looking at you, right? And why are you taking my carcass? Okay, so we know that this confrontation scavenging is happening also now in certain hunter-gatherer groups, but this is a case where information exchange about whereabouts of the resource, in that sense, it is similar to language, by the way, but it didn't get stuck at level. And the other one is highly cooperational, because if you don't cooperate, first of all, everybody sees you and you are in the group. So if you don't cooperate immediately, somebody will punch you on your head. And the feedback is practically immediate. Okay, that's a story. I don't want to go into this. I could, but I want to mention other things as well. So amazingly, about a year ago, this paper appeared from Klaas Anderson and Peter Turnberg, and the title was "Toward the Macroevolutionary Theory of Human Evolution, the Social Protocell." This was actually a very surprising. I got it for refereeing. Now the title is a bit misleading at this stage because there is no mathematical model yet behind it. It's a qualitative argument, but in that way, or semi-quantitative argument. But in sense, it's very detailed. It's freely downloadable from biological theory. So they are actually comparing what you saw, the stochastic corrector model, with the kind of growth and fission of primate groups. Now, this kind of growth and fission of groups does happen in chimpanzees and also hunter-gatherers, but it's a surprisingly under-researched process. But it's very important to this argument because what they are saying, I hope I can explain it very briefly but still informatively, is that what you are seeing here is actually a network of social interactions and especially what is very important, social knowledge. Right? So now, of course, how fast, how efficient this growth is going to be, that will have an effect on, or that will be depending, on what kind of cultural information the individual members of the group are carrying and how efficiently they can transmit it to the next generation, right? So it's similar to the dependence of this protocell fitness on the gene content. Here, it is mainly cultural content that is affecting the growth and division efficiency of the social protocell. So they maintain that the real unit here, because individuals can be replaced, but here the real unit is at the social level, this network of, if you wish, co-adapted mean complexes. Now, don't kick me, Eva. I know that some people hate the word mean, but I want to be very telegraphic. I'm saying it's the co-adapted mean complexes that are important for this kind of process. And if this process carry on, it will be more and more co-adapted this time. Right? The other thing I want to emphasize that nobody thinks that this drives our society today, but the proposal is that that stage was a critical thing into the transitions to human type of society. Although it also comes with other things, as we know, as opposed to the chimpanzee societies, at this level of hominization, what you have is the appearance of reproductive leveling, right? Because in the hunter-gatherers, the reproduction is, there is food sharing, reproduction is practically evil, that's not evil, it's even. And when it becomes evil is actually when you get agriculture. Then you again have a distortion in basically anything because of property. But here not so. But also when looking at the fate of the cultural content, you will see that although there are different packages of cultural information, but the information that is necessary in order to process food and the information in order to deal with people who are sick, You cannot replace one with the other, right? So from a cultural point of view, you have the sociant, and the origin of the sociant is actually an egalitarian type of transition. If it's an egalitarian type of transition, you cannot expect anything like reproductive division of labor in the cultural sense. Right? Now there is, okay, I explained this, there is cultural, there is ongoing work that is now doing the modeling of this, because of course you must also model, the cultural transmission of information within the communities and so on. So technically this will be involved. How many minutes do I have?
Go ahead, I'll tell you that.
Okay, all right. So now, of course, this begs the question. All this begs the question. You see the kind of origin of language, the efficient sociant and so on. I mean, they require excellent. They require efficient brain power. I mean, you cannot explain this away. We know, for example, that there are even cells in the human brain that are specific to the human. There are new cell types even, but also dynamically speaking. So you take a piece of cortex from a living monkey, and you take a piece of cortex from pole. It can be done, like later I explain you during dinner how it is being done, and under what circumstances. You take it, and then you make tissue culture, and for example, you trigger one neuron with a monosynaptic event, right? And you will see that the avalanche of activities is much more complex in a similar piece of human cortex than in the brain of the macaque, right? So obviously there are innate differences between the processing capacities of the brain, and the question is, what are the elements of that? We had a go at it, and we are still having a difficult go at it. And that was the question, for example, by Santiago Ramon y Cajal's extremely famous neuron. So with Crisanta Fernando, we were exploring for a long time. Now he is actually with DeepMind at Google. This question, can you actually imagine that you are sustaining a Darwinian type of natural selection in real time in the brain when you are, for example, learning language, or you are solving complex problems like general relativity that took 10 years for Einstein, for example. Developing language also takes several years. The question is whether this kind of adaptive complexity, one of the mechanisms behind that might be per analogian, a Darwinian type of dynamics. And now you can remember the case of the other transition to the vertebrate immune system. We don't know the answer, so don't get me wrong. It's very hypothetical. Let me just mention in passing, I cannot resist. It's so beautiful and it's so annoying because I could have discovered this. I have a good list of these things, right? So this is one, unfortunately. So it is from Harper, and there is Chalizi, but Chalizi's paper is incomprehensible. I cannot understand it, but this is very clear. So here you see the Bayesian update equation, and here you see the discrete type replicator equation. And here is the translation table. It's really infuriatingly, it's a penetrating insight. And what is important, of course, this science is trivial because every gene or every organism, depending on what you are focusing on, is a new hypothesis how to do life well in that environment. If it's a bad hypothesis, it's going to be pushed down. If it's a good hypothesis, it's going to be pushed up, just like different forms of Bayesian hypothesis. Okay, but of course, that means, by the way, that natural selection, Darwin natural selection, is inherently Bayesian in that sense. But mind you, it's also true, unfortunately, that the fact that we know that from experiments that the brain is doing Bayesian type of approach to many problems doesn't mean that the algorithm is Darwinian, because you could do it by other algorithms, you know that. The question is, how do you do it? Probably in many ways, depending on what problem you solve. And here is one of our recent attempts, whereby we actually designed the mechanism whereby you have got several neuronal layers, and these so-called dynamical attractors, I don't explain this now, I know, but time is limited. So these are attractors within the network that actually can carry the information. So if you are provoking this network and repeatedly you are feeding the information that you get out, and you have an evaluation step whether what you find is good or bad, so you can actually find surprisingly fast a good solution in the existing brain if it's already there. If it's not there, what you do is you have to introduce novelty, right? So you must generate a novel thing, and this novel thing will carve out a niche in the existing neuronal population. And once you have it, this new attractor can serve as a stepping stone. So from you, then you can solve the problem from the provocation right to the end result of the solution. But there is a crucial element. It's copying. This is what Change, Edelman, and the others didn't have. That's the, I mean, you can, I must I must tell this because it's so fantastic that a disciple of Edelman told this to him, that, you know, unless you have some kind of replicator dynamics, you don't have the full capacity. And then he said, yes, but I hate Richard Dawkins, I don't want to. And so now we don't hate, although we don't agree with everything. So ultimately, and that's the final slide, this is under publication, we have the first evidence from cultured cells that if you train a group of neurons, it's fetal neurons in a petri dish, and then you record the spontaneous activity after training, you channel it into a naive group of neurons of the same kind, but they were not trained. Yeah? It listens to the spontaneous activity. And then you make the trial, and it knows. Okay? So the information has been transferred, but we don't know how. Okay? But that's the first experiment that shows that this copying is somehow there. According to our intuition, it shouldn't be there, but nobody knows yet why this is the case and how it can be generalized. So there was still made by for labs, don't get me wrong. But if not, that will be probably the most, I think so, the most important application of Darwinian ideas beyond the original context, because then it explains a lot. Oh, thank you very much.
You talked a few slides ago. You talked about a situation where a major transition sort of started and got stuck, and then you have situations where it starts and goes all the way to a situation where you de-Darwinize everything that happened down below. But then there are all those situations where you see Darwinian effects on both levels and they kind of interact. And it seems like in the human case, something of that type is happening. Can you say a little bit more about this complexity of two levels together, each working according to its logic, and then the relationship between them?
Well, the answers to these questions are usually best given within the context of a model, because then you get a quantitative effect. Let me just say that there is this old description, now old, by Hamilton, where he used the approach of price to describe multi-level selection. And the question is, you know, what part of the variation is relevant for, you know, for the dynamics at the lower level, and what part of the variation level is important for the higher level. To my mind, it's a limited thing because it's not really dynamic, right? It's not dynamics in the sense that a physicist would, or even I would understand, but it's important to have these levels. So I think it is true that in the case of human evolution, of course, we went up, to that kind of thing. I don't think that we went up consistently, right? So occasionally, we went down. And from the very technical point of view, actually, I don't think that we have ever been superorganisms, right? Definitely not biologically. Culturally, it was sometimes, especially in this period of the social protocell, if it's true, then that was quite close. But even it was a transient thing, as we know. Now we are much more similar, if you want to put it like that, to the so-called ant super colonies, right? Because we have lost many things that would give us, although it might come back. So some people expect that the ant super colonies will collapse. And if you look at recent human history, what you see is renationalization of politics, right? So it might be that it's not by miracle, that's the nature of the thing, right? And as I told Eva, in Israel here, I feel very much at home, right? I mean, if you look at Hungary and Israel, okay, sorry, but it's funny.
Thank you very much. So it reminded me that there's a lot of valuable material here, both in our talks and in our slides. So we would like to have them all for the benefit of this group to be on our website. It may also be for the benefit of many other people that are interested in these topics. And so we would like to have both the talks that we brought, as well as your presentation in the format of a PDF, put on our website. If any of you is not happy with this decision, please let Tamar know, and we will act accordingly, of course. That's what we are going to do. So our next speaker could not make it today to be physically here with us. We are very sorry about that. It's Dan McShea from Duke University. However, we take comfort in the huge amount of carbon that was saved by this decision. And we are going to have him, after all, thanks to Tamar and the Zoom technology. Is he with us now?
Yeah, with us. Dan, can you hear us?
I can hear you. Can you hear me?
Yeah.
Yes? No?
We can hear you.
Great. Okay.
Yes, hello,
Shall I-
Dan. You were already introduced.
Oh, okay. Shall I just go ahead and start?
Yes, go ahead.
Okay, well, thank you all for the invite, and thank you for setting up this electronic participation. I wish I could be there in person. If I need to talk louder or softer, please just let me know, or adjust my screen somehow. I like to start my talks when I can with a quote from Mark Twain, and I've got a good one for this talk. Twain said, "Get your facts straight first, then you can distort them as much as you please." And that's pretty much the structure of my talk today. In the first half, I'm going to say some things that I at least hope are true, some facts, and in the second half, I'll distort them to create skepticism about the major transitions. I'm going to start with a trend in what I call hierarchy. And the slides are not switching.
Are you sharing?
I am sharing. Do you have any slides at all up there? What do you see anything at all? Hello?
1 minute, Dan. Yeah. Now we can see it.
You got the first slide? The
Yeah.
trend in hierarchy?
But it's not on presentation mode.
Right. How's that?
Much better.
Yes? No?
Perfect.
Okay. So this is a trend in hierarchy, and interestingly, it's one of only 2 trends that have been documented quantitatively at the scale of the history of all life, and I'll show you that trend in just a minute. It's similar to, maybe identical to, the trend in individuation and levels of individuality. It doesn't include all the major transitions, it's just those that represent a trend in hierarchy. So notice the levels here: prokaryotic cell, eukaryotic cell, multicellular individual, colony, and I've added a possible 5th level, colonies of colonies or super colonies. Just be aware that the trend is open-ended. In principle, it's open-ended, that is. We could, in principle, have levels 6, 7, 8, and so on. And the question I want to address here is, where do people stand on this scale, and mainly consider where between levels 3 and 4 we stand, although the possibility that we occupy level 5 is constantly being raised by people I talk to about this. So 3 preliminary points before I get to my reasons for skepticism. The first has to do with my notion of hierarchy. I have a purely structural view of hierarchy. Here is what I call a thing, an object. And what you're supposed to understand here is that it's a collection of elements. Those are the little circles. And the arrows show interactions among the elements. And a thing is understood as a collection of elements that are highly integrated internally, highly connected among themselves, and less connected externally. So you see lots of arrows within that big oval, and very few arrows that penetrate the oval to the inside, going either in or out. That collection of connected elements is a thing. Most things are objects, or many things that we like to think of as things are objects, but they don't have to be objects. They don't have to be bounded. So here's hierarchy. Hierarchy is things within things. You can think of it as objects within objects. And what I've done here is I've highlighted 3 sub-things, 3 parts of the whole. And they too represent collections of elements that are relatively highly connected with each other and less well connected outside their own sets there. I hope you can see that. And now I'll go back a slide, and you can still see the things. They're not bounded in any way, but if somebody asked you to pick out the clusters of connectedness, you would probably pick out roughly the ones that I circled before. Important point here: thingness, degree of internal integration and external isolation comes in degrees. You can have degree of thingness, you can have degree of thingification. I don't know what the right word is here, thingatudinosity or something like that. So here's hierarchy, things within things. Preliminary point number 2. That was a purely structural notion of hierarchy. I'm going to now introduce selection and use it to move to what I call degree of individuation. And everybody means something slightly different by individuation, so let me tell you how I'm using it. We're starting with, on the left, say, a eukaryotic cell. And when selection operates on the cell, let's say to increase size, it doesn't have to be for size, but it could be. What you might get is aggregation to form a higher level whole, but it's just an aggregate. And that's a stepping stone along the way to the next highly individuated level. That first stage, aggregation, is just a stepping stone. If selection were to keep acting on it, say for efficiency or something like that, we might get division of labor. And that second level, B, differentiation, is trying to show an organism, a higher level organism, meaning a multicellular quasi-individual, near individual, with 2 cell types. Okay, that's the second stepping stone. The third is what I call the tissue or organ level, or anything with intermediate level parts. That's whenever there's spatial structure that arises in connection with the differentiation. So I've got 6 of those cells arranged in a triangle, and 3 of them as a little cover up top. That's sort of tissue or organ level differentiation. It's spatial organization at a level above differentiation. So we've got a scale here that goes from any level, and I've been talking at the cell level, but it could be at other hierarchical levels. And selection acts, producing a trend from left to right through various stages: aggregation, differentiation, the tissue and organ level, And then the next step up could well be to the next full level, the next level of containment, the next level of nestedness of parts within wholes. This is just one possible scale for degree of individuation. There's a lot of possibilities. Yochai and Ayelet have developed another one. There are doubtless others out there. They're useful for different purposes. It would be good to have a meeting of some kind of conference on individuation scales, if such a thing were possible, and talk about their differences and similarities. I've thrown on some examples here just to make it more concrete for you. There's our single-celled solitary eukaryote down at the bottom on the left. Level 2C, I call it. It's highly individuated at its cellular level. Moving to the right, a multicellular alga like gonium, which is has only one cell type, and there's no strong spatial organization to them. The next level up is Vaucheria, which is also an alga with two cell types, two specialized cell types. And then at level 3C, we've got an enormous number of organisms ranging from volvox to cats and dogs and trilobites and so on. Things we ordinarily think of as solitary multicellular organisms. There's a lot in that category. And then the next level up would be an aggregation, a simple aggregation of these at level 4A. So I'm just introducing you to my scale here by showing you some organisms and where they would fit on it. While I'm here, I can't help but show you the trend at the scale of the history of life in these things. On the x-axis is time in millions of years. So this is the entire history of life going back 3.5 billion years. On the y-axis is hierarchical level with my scale of individuation mapped onto it. We start at the bottom with a full-blown, highly individuated prokaryotic cell, a bacterium or an archaebacterium of some kind, and then degrees of individuation of eukaryotic cell all the way up to modern protists. And then multicellular, 3A, 3B, 3C, up to fancy multicellular organisms. And then the colony level. And for each one, the data point you see, the little square, shows the first occurrence of that level in the fossil record. So my scale of individuation was designed with the fossil record in mind. I was going to be looking to create a graph like this. I had to use criteria that were physical, phenotypic, and would appear in the fossil record. And this is the trend in first occurrences in hierarchy over the history of life. Let me strip out the names so you can see the trend. There's a couple things that are fairly remarkable about it. One is the huge acceleration that you see in that curve as it swoops upward. It took forever to get from a simple cluster of prokaryotic cells up to full-blown eukaryotic cells, 1.5 billion years. And then throughout the Proterozoic, things continued to accelerate until we get to just before the Phanerozoic when it really took off. So point number one is that there was a huge acceleration here. Point number two is that there seems to have been, at least according to the fossil record, a very abrupt truncation. That last data point you see at the colony level 4C is bryozoan colonies from 480 million years ago. And we have nothing in the fossil record at the supercolony level. Let me just remind you, level 4C is not just any old collection of multicellular organisms, it's a highly individuated one. Bryozoans have multiple zooid types, and each zooid is a multicellular individual. They also have differentiation among those zooids, and they have the equivalent of tissues and organs at the colony level. And I could explain those if I had more time, or we can go back to it if anybody wants to hear about it. Okay, so that's the trend. The question question one could ask, and that I'm not going to linger on here, has to do with acceleration and the truncation, but there's also the question of where humans would lie on this scale. Are we at level 5, or even at level 6 or 7, or are we down at level 4C? Standard thinking, by that I mean what answer you get if you ask an undergraduate, is that humans are at level 4C. We are a highly individuated colony. I'm going to argue here in just a moment that we're more like 4A. We're just a simple aggregate of multicellular individuals. Preliminary point number 3, there's something that I call a complexity drain that Dr. Herrera has actually talked about in different terms, but it's very similar. What I have to say is very similar to what he has to say. As you move up a level, and in this case, I'm showing you what happens as you move from the solitary eukaryotic cell stage to a multicellular individual. As you move up a level and the parts differentiate, selection favors the streamlining of the parts in the interest of efficiency. So if I'm a solitary paramecium or amoeba or something like that, I've got to do everything for myself. I've got to defend myself. I've got to reproduce. I've got to feed. I've got to move around. If I'm a skin cell in a multicellular individual, food is delivered to my doorstep. I don't have to defend myself. Oxygen is delivered. Selection is going to favor the streamlining of cells in multicellular individuals to reduce the number of parts. And this shows an extreme case of that. There's a paramecium, which in this picture appears, that was how I picked the picture, like it's got a lot of parts going on within it. And over on the right is an extreme case, human red blood cells, which have no parts within them. Well, very few cells actually fall at these extremes. I have a quantitative study that I've done where I compared a large number of taxa, and I can demonstrate quantitatively a decrease in number of part types comparing free-living protists to cells in multicellular individuals. Our cells are stripped down, and that's what I call the complexity drain. That's at one level. That's cell going to multicell. There's some reason to think the same process operates at lower levels in the move from solitary eubacterium to a part within a larger whole, namely a mitochondrion. We at least know that a number of genes has been stripped down considerably. I think it likely, but I can't prove it, that number of other parts has also been reduced. In the transition to coloniality in bryozoans, the picture you see on the top right is of monobryozoon. It's a solitary, one of the only living solitary bryozoans. I've done part type counts within monobryozoon and also within a zooid in a highly individuated colonial bryozoan, and monobryozoon has more part types than an average zooid in a colonial one. This is impressionistic. I don't have real data to go with this. I would like to get some. Let's just assume that the principle is valid for the rest of the talk, that there ought to be, at least, that selection should favor a stripping down of individuals as they move into the larger whole. Okay, finally, reasons for skepticism. I've got 2 questions going here. The first one, question number 1, how high do human societies reach above the colony level, level 4? Are there super colonies and super colonies and super colonies? We don't have words for these things, probably because we don't know of any others in nature. And my argument is going to be no, we're not at level 5, 6, or 7. And the reason has to do with what I call cross-cutting interactions, which undermine hierarchy. So here's my diagram from before showing things within a thing. And what we've got is a bunch of isolated, semi-isolated elements that are highly connected amongst themselves, and the hierarchical structure would be clear even if I removed those helpful blue dashed lines. But here's how it actually works in people, and I'm going to give you an example that starts with me. I'm the black dot sitting up there in the upper left with the big inside the family circle. That's my family circle, and I exist within it, and I'm highly connected within it. Turns out my connections are much broader than that. I'm also a member of the Duke Biology Department. I'm also a member of the Tennis League. I'm also a member of a volunteer fire department. And this is going to be true for every single human individual here. If you can, in your head, mentally draw arrows from every element here, cross-cutting to every blue circle, and now add a lot of blue circles, because this is just 4 possible roles that I could fill. You're going to get a massive arrows going every which way, and those lower level units are going to disappear. In other words, speaking in general terms, cross-cutting interactions undermine hierarchical structure. There isn't going to be much hierarchical structure left after you draw all the actual interactions that occur. And I think this is the case for human societies at all upper levels. We're pretty good individuals. I'll talk in a moment about whether we're pretty good societies, but at super societies, super societies, I'm suggesting there's just too much cross-cutting interaction to preserve hierarchical structure. Question number 2. Granted that we're colonies, how individuated are we? And here's the scale that I showed earlier. In this case, over on the left, what you see is a solitary human individual, and then an aggregate at the A level of human individuals, and then a differentiated aggregate of human individuals. Well, just what is our commitment to division of labor? I think Dr. Herrera and I could have a good debate about this, but here's my impression anyway, and this is just impressionistic. We're not very committed as far as division of labor goes. We don't have phenotypic castes. We don't have permanent divisions, morphological divisions that identify people by what their specialization is. And we can switch back and forth. So for example, I'm a decent professor, I think, I hope. I'm a terrible carpenter, but I could become a carpenter. And in fact, I have a colleague who made that transition from being a faculty member to being a carpenter. It took a lot of training. It wasn't easy, but it could be done because the commitment, the degree of specialization just isn't that great. We don't have that much serious division of labor. Actually, my parents back in 1948 ran away from New York City to start a subsistence farm and learned how to grow their own crops and build their own chicken houses and provide their own food essentially. And they did it from the ground up. They were city people. But because there was no morphological commitment, no phenotypic commitment, no deep psychological commitment to being the city slickers that they had been, they could undo all that and start over. And that's not true of, let's say, zooids in a highly differentiated bryozoan colony. I don't know much about social insects. I suspect it's not true. I believe it's not true of ant castes. So again, my position here is that no, there's not much commitment to a division of labor, not compared to what we see in multicellular cells within multicellular individuals. Again, the same question, what level within the colony are we at? And here I'm gonna argue that the complexity drain has been pretty negligible in people. We haven't lost organs, we haven't lost tissues, we haven't lost cell types compared to solitary multicellular individuals. Some hypothetical ancestral primate, compare us to them. Yes, there have been some reductions of various sorts, like the shortening of the intestine and so forth, owing to changes in diet. So it's not like we haven't proceeded at all down the complexity drain. We have, but it hasn't been that substantial. I tried to come up with, and I'm not sure I was successful, a graphic that would depict this, but unfortunately it depends on your knowing something about Star Trek, The Next Generation, that old series from the 80s and 90s. There was an entity called the Borg, which was a highly individuated collective in which the individuals were stripped down automata, unable to have their own independent thoughts, much less behave independently. This is cells in a multicellular individual. In fact, that analogy was used in this science fiction drama, and we're nothing like that. Thank goodness we're nothing like that. Okay, same question again. Are we level 4C? Are we something simpler like 4A? Are we a simple aggregate or a highly individuated collective? And here my suggestion is that we're not because, or we have reason to suspect we're not, because the wrong sort of natural selection has been acting on human societies. Selection acts in 3 modes, not the usual one that's explained in the textbooks. There's a stability mode. Any organism that can be around longer than its competitors, it doesn't have to out-reproduce them. It doesn't have to reproduce at all, in fact. If it can just survive while others fail, it's considered selected. Then there's selection in the growth mode, in the expansion mode, and clonal organisms are especially good at this. In fact, the famous fungus, Armillaria, that's the largest in terms of area, not in terms of mass, obviously, but in terms of area, the largest organism on Earth, grows clonally. And selection acts on it in the sense that if it can expand faster than anything around it, again, reproduction not required here, just expansion, then it is considered selected for. And then finally, there's differential reproduction, which is the textbook version of it. Thing is, differential reproduction is how you get complex adaptation. The other two modes of selection are just too slow to produce anything but things that grow fast and things that last a long time. And human societies seem, to me anyway, to be selected almost entirely in stability and growth mode. Societies displace each other by growth. They persist where others around them fail, but they don't reproduce that much. Fissioning is fairly rare in society, at the human social level. And that's the kind of fissioning that would be necessary for there to be differential reproduction, that would be necessary to produce complex adaptations. So I think, yes, that's the end, and I'm available for questions.
Thank you very much. Questions?
It's about cellular labor specialization. We don't need to be different genetically to be to specialize in different labors. As a matter of fact, the first cell, the cycle, is pluripotential. So it specializes by means of methylation. So a similar mechanism can go and follow the specialization in the human brain. As a matter of fact, labor specialization, some activities, mental activities, are specialized because of brain plasticity at two levels: the molecular level by methylation, and the neuron plasticity, dendritic tree. So we don't have to be genetically different to specialize. Methylation by some means, by maybe some point mutations that conduct. I don't know. We don't know yet how methylation occurs between environment and the cell, but we know that environment influences methylation in cells. We don't know yet how it does it, but yes, it influences. So a sort of evolution of this type can go on in the human societies. And another thing is this cutting from the groups, the interrelated groups. You said that you belong to a family, but you belong to a club, and you belong to a scientific society, etc. But that's going on in cells too. A cell can be, for instance, an epithelial cell, and it can have other activities, such as immunological activities. So it doesn't have to belong exactly to one, do just one job. It can belong to multiple organs, multiple systems. So I'm arguing with you about this. I think it can be. We can specialize. We can have labor specialization.
Did you hear the?
So I can't see who I'm talking to. Who was that?
Edwin Pass.
Okay, thank you. The first question I'm a little puzzled by, because I never talked about genes and nothing in any of my complaints. That helps. Thank you. Nothing in any of my reasons for skepticism had anything to do with genetic transformation. I'm happy to have plasticity cause it. I'm just saying that nothing has been permanent. None of the specializations that have taken place in humans have been permanent, even within a single lifetime. It may feel that way. We may feel like specialists, but and maybe as we get older, we become more and more committed to a particular way of being. But retro transformation, moving back to a generalized state, is pretty much always available in a way that it isn't for most cells in a full-blown multicellular individual. And then, what was your second point? I've lost it.
A human being can belong to various.
Yes, right. The cross-cutting interactions. Yeah. So my point is that this is an expectation. The more and more cross-cutting interactions there are, the less and less you expect to see any hierarchical structure. And I want to add to that also that in real multicellular individuals, like us, for example, a liver cell is a liver cell. It doesn't you know, have a night job as a kidney cell. There is a strong commitment to being within a particular tissue or organ and functioning within that tissue or organ. So there are interactions among the parts. Of course, the multicellular individual couldn't function otherwise, but the strongest interactions tend to be within the tissue or organ that it's functioning within. And if you were to draw an interaction map of a human individual, physiological interaction map, including physical interactions, you would see strong hierarchy, I think. I'm not sure if you were to draw a complete map of a human society and all the interactions among individuals within it, you would see the same hierarchical structure. This is a graph theoretic problem. I don't have numbers. It would be nice to see numbers on this sort of thing.
I think I agree with you that at the level that the type of transitions that you describe, which are nested hierarchical transitions, you cannot place humans at a very high level from this point of view, although I'm not sure if it's 4A or 4B or whatever. However, these are not the only kind of transitions that exist. And there are the kind of transitions, for example, the transition from RNA as both replicator and enzyme to DNA, to the division of labor to DNA, RNA, protein. There is a transition into neural organisms when you have a new communication system. And there is a transition to language. We have something that no other, we human beings have a kind of system of communication and representation which is unique, is symbolic. It's open-ended, it allows open-ended evolution, called cultural evolution. It leads to a new type of selection. We have new value systems, which are abstract value systems, and so on and so forth. One can talk a lot about it. So if you're looking at transitions, not only as hierarchically nested transitions, but you're looking at transitions which lead to a new type of information system, information transmission, and a new type of representation, and a new type of developmental selection, and new modes of selection, such as open-ended cultural selection. I think that if you are looking at these transitions, humans are absolutely unique. And the transition to humans is very much a transition, a major transition in evolution. Not for nothing. I think that it is not a coincidence that people who are talking about the transition to humans are very often focusing on the symbolic system, especially on the linguistic system, because it allows this enormous jump in organization, internal organization.
Yeah, I tried to, maybe I wasn't clear enough in the beginning. I'm only talking about transitions in individuality. I don't pretend to even understand what a major transition is, and my trouble with it is that it's combining two separate sets of criteria, one having to do with information transfer and language and so forth, and the other having to do with individuality. So you obviously want to add something, and I can't hear you because you don't have the microphone, so could you pass her the microphone?
Individuals. We are different types of individuals, not because we have more hierarchical, because the society has more hierarchical levels, but because the way our mode of being, our symbolic mode of being is different. Our self, the way that we construct the self psychologically is very, very different. It's culture dependent, learning dependent in a way that is very, very different from other animals that of course also have. So it, again, depends very much how you think about individuality. And if you're thinking about individuality, again, in terms of hierarchical nestedness, yes, you're right. But there are other ways of thinking about it.
Yes, of course. There are other ways of thinking about them, and I'm open to talking about them. I picked the ones that I could operationalize, and maybe other people have operationalized what you're talking about. I don't doubt that people are different from, every species is unique for one thing, but I'm also very impressed by language and what our culture has wrought that no other species seems to be able to match in 1,000 different dimensions. But I've picked something I can operationalize, degree of individuation in a structural sense. And now I'm just sort of, the attempt anyway is to blindly map organisms on Earth onto it. And at the end of the day, and I think you sort of agreed with this, using that scale, we just don't see much individuation. I'm not denying there are other scales.
I think, yeah. Let me ask a question. Actually, perhaps try to ask for a clarification.
Yeah. Hi.
Okay.
Go ahead.
That in your classification, in level 4A, all the
Yes.
series in the graph were very similar, were actually identical. Okay?
Yes. Yep.
And this is not the situation that the modern society is. Even though I agree with you that you could become a carpenter, but in fact, most of your life you devote to a single occupation. This is very different from the case where all of us conduct the same set of functions, okay? In contrast, our modern society is highly elaborated, okay? Each person performs a set of functions which are different than the other persons in the society, and society is so much more complex than, take for example, human situation 20,000 years ago. So I guess it comes down to how you define division of labor. I agree that like stem cells, we can still have the potential to switch occupations, but division of labor is certainly there, I would argue.
Yeah, and I may have said the opposite. I didn't mean to say the opposite. What I mean to say is the commitment to division of labor isn't there. We don't see phenotypic casts, and that's one of the reasons the reversals can happen. So no, I'm not denying that division of labor exists. It's just that if you were to compare, pick a species to compare us to, and I'm not an insect person, maybe somebody in the room can help me out here. There's something called temporal polyethism in bees where they switch roles as they proceed through life. They move from one functional role to the next. That's the level at which I was imagining human societies operating. They do specialize, but they're not as special. The commitment that you see in ant colonies just isn't there. Do you have a comment from somebody who knows what they're talking about? I don't hear anything. No, I don't hear anything. Ah, wait, try now. No, sorry.
Now it works? Okay, good. So
Yeah.
first of all, I think as I gave, I explained in my talk, for the humans, it actually matters a lot in which phase of history we are talking about. The social structure has been evolving from the biological point of view as well as from the cultural point of view. So I think that you have a different picture depending on what phase you are in. It's a general comment, but a more technical comment is the following. So imagine, it doesn't, sorry, this now, okay. So
Now you can hear him, Dan?
Yes. You're right in front of the camera.
Okay, excellent. So that's a technical comment, right? So if you take the structural hierarchy approach, right? Then and if you look at the drawing of what you gave for the human society, right? Then you see that the number of links between the different individuals is actually increasing. What you have in the end is that you have one unit, right?
Yes, exactly. Yes.
Okay, so that is in contradiction in a way to what you were suggesting, right? So one could argue in the opposite way, that these cross-cutting features are actually making the new unit much more dense and much more interwoven than any time before.
Right. I was addressing the issue of whether there's levels 5, 6, and 7, and so on at that point. And my only point was that interconnectedness tends to undermine hierarchical structure. Your point is also valid, or could be valid. It's an empirical question. It's a graph theoretical question. How much is the connectedness now? How is it changing over time? How does it contribute to individuality at the level of the whole planet? Is that what we're talking about? And the answer could be enormously, for all I know. Yeah.
Thank you.
By the way, this reminds me of an example. I'll just do this quickly that I like to use. You want to find individuals, human individuals that are more stripped down, more committed, less interactive. Turn to the Industrial Revolution in any Western country, take Britain, where we had factory workers working 14, 18 hours a day. Their lives consisted of eating, reproducing, and working, putting heads on a pin in the paradigmatic case, and that was their existence. So we're capable of that kind of commitment, but through most of human history, I don't think you see it. Okay, I just wanted to add that. Thank you.
Would it work? Hi. I think that what's happening here, and I think it's been happening in some of the talks before, is that when we look at the question that we're dealing with at different levels, we get pretty unified answers about certain transitions, like at the cellular level, or in some of the insects that seem to have finished the process. But we're getting very different answers with respect to humans, depending on where we're looking from. And I think that if we look from your perspective, what you were saying, I totally identify with what you're saying. The results are not very impressive. But if you look at the point of view of information sharing, of the question of, for example, what is the unit that is going to be arriving at conclusions in this workshop, then I think that in a certain sense you're going to say there's a colony, a super whatever you want to call it.
A temporary one.
Sorry?
A temporary one, yes.
A temporary one, yeah, there are differences. And I think that, you know, from my own work on language and what Eva has been saying, the major transition in our case most dramatically has affected information sharing and the way we reach conclusions, not individually, but through constant conversation with others. So maybe what's happening, and this is also related to the question I asked you before, maybe what's happening in the human case is that we're somewhere in the middle of a certain process that if we look at from this perspective, it doesn't look very impressive, like you said, but we look from the other perspective, it looks like a very, very unique thing. Maybe there's something more complex.
Yeah, all I can say is because I'm so blinkered when it comes to talking about language and information sharing and that sort of thing. I don't know how to fit it into my conceptual scheme at all. It sounds like two separate conceptual schemes, two different books, right? And until I can't really comment on it except to say, yes, I'm impressed by what people do in all kinds of ways, but is it individuality at the colony level, right? That's what I always want to keep asking. Is that's what it is? However impressive it is, however unique, however interesting in all its different ways, is it what we're talking about in the case of cells going to multicells and bryozoan colonies forming? And my answer so far is, it sure doesn't look like it. Something interesting, but not that.
Actually, it might become that way. You start with an elaboration, with an elaborating within the society is not individuation at all. And all of a sudden you have all the potential to be realized to become the society, to become one large individual. So it might be a pre-adaptation.
Yeah, so the notion of a global individual using what I consider the standard criteria, my standard criteria for individual, that's not out of the question at all. That's entirely possible. I need numbers. we need to get quantitative about these things.
Thank you so much.
Oh, thank you.
Hi.
Hi.
I wonder whether this scale of that you use could be in a way used for the description of the human brain. We look at it as a super colony.
I need you to continue because I don't know enough about brains to say. It sounds promising.
I think that we can compare the human evolution as an organism with the evolution of multi-organism by the ways in they communicate, the elements communicate within it. And I think a good approach could be nervous systems. First nervous systems were networks, and they evolved to have ganglions, and then they evolved to have brains. And we have seen pretty much the same evolution in human communications. Human communication systems, first, they were from people to people, person to person, such as chemical communications in the multicellular organism. And then we evolved for far communication, long-range communications by means of electromagnetic communications, telegraph, and then the internet. But we are still a network with no ganglions. But we can predict that some sort of brain is going to form in the future. Yes, it's possible. It is possible. A brain it's a computer, needs inputs and outputs. And these centers of processing can have huge inputs from the whole world, from a city, from the Internet of Things, from the Internet, and sense and make decisions. For example, how much of some product has to be made following the offer and demand. For example, that's an example. So there are many things that a huge brain, a huge center of processing can do that we don't see that yet. We see some nodules such as Google and Wikipedia and these portals, but they are still small. If we follow the evolution of nervous systems, maybe we can have some insights in the evolution of communication systems and centralized information in humans.
So hierarchy.
For example, another example: the cell has all its information in the DNA. All the information of the cell is in the DNA. But the cell has to silence much of this information in order to specialize. The same in actual societies, in modern societies, we have all the information available in this cellular phones. But we don't use that information, we use just the information we need. We have to silence the rest of the information. So I think if we speak in terms of information managing, we can compare the multicellular organism with the evolution of humans.
So let me just say that while I can see the hierarchical structure of the brain and its individuation, multiple hierarchical levels, specialization of regions, specialization of cell types, all that makes enormous sense to me. One of the reasons it makes enormous sense to me is that we know selection has acted and is acting at the level of the individual. And while the analogy, the picture you paint is an attractive one, and the analogy seems like it could be at least a good one, I don't see what selection pressure could be driving it, not in evolutionary terms. There could be internal dynamics to human societies that drive it, but I can't see any selection pressure that drives it because societies don't reproduce. There is no selection in that sense at the level of the colony, or little selection in that sense. But go ahead, show me wrong. I'm happy to be wrong.
Yes, I'm not thinking in biological selection, but
hang on, you're breaking up. I can't hear.
On a higher level. For instance, structures, goods in a society are manufactured because there's offer and demand. That's not biological. And there is a competence of products in the society, and it's not biological. It obeys to offer and demand. So offer and demand acts like biological selection. There are other considerations at population level that are different, are not biological, are cultural or sociocultural. So it could be a selection, not natural selection, but artificial selection in the mean of a pressure in the market, for example.
Well, all right. I'll just say that I can see selection at the alternative hypothesis, selection acting at the level of the individual could easily produce all of this. Corporations producing innovations, individuals producing innovations, communicative expansion of communicative networks, all this could be driven by individual initiative. We don't need any higher level process to account for it. I'm not denying that there could be higher level processes. I just don't see the evidence for it.
I just want, hi, can you hear me?
Yeah.
All right. Just general comment. I guess we will not end this discussion, but we just we'll probably continue this in the next few days because it's raised the question of what is a transition. Because I feel that what Dan was trying to do is to say, look, there are certain criteria of what a certain way by which people view transitions, and according to this criteria, I don't think that humans, recent evolution, is qualified for this. But if you think that transition can be any evolutionary change that is major or significant or important, not any, but exactly the question is, many people will say, okay, this development is a transition, and where is the line to define a gradual evolutionary change? At which stage we say transition is a transition? Yeah, some of it is really fine. Mm-hmm. Yeah, I don't expect an answer now, but I think this will be part of what we. Exactly, that's what I mean. Yeah.
I can't disagree with what you said. Sounds right.
At this point, I'd like to thank you very much for your talk and for joining us so early in your morning. Actually, in the evening, I guess. And hope you stick around.
I will be looking in on this as much as I possibly can. This is a really interesting looking meeting. Thank you for having me.
Thank you. And now we have a few minutes, just a few minutes before the is delayed.